Okay, let's continue for the last bit of biology. So we reminded ourselves by now about energy consumption and vascular system, and now we talk a little bit about cerebral blood flow, so that we know a little bit about what's going on in terms of blood flow, not only the anatomy of it. So what is blood flow? Blood flow is the volume of moving blood per unit time. So it's actually a physical quantity, and is, of course, if you think about the regulation of blood flow, it has to do with how much pressure is applied. So what the pressure that is generated by actually your heart beating and the constriction of the arterial system, and the resistance that is applied to that in terms of the constriction of the vessels. So one thing is that the resistance of a vessel depends on the vessel diameter, which is kind of intuitive. So if you think you have a tube and you apply some water and then you make the tube diameter small at one point, then of course less water will run through it in unit time, and you have less blood flow. So if you have in mind that the heart is beating and there's kind of this pulsatil flow in large vessels, there is this, or what's this name? The name of this thing? There is basically, in terms of the physics of the vascular system, you can show how things are set up, that this pulsatile flow in large vessels, due to the beating of the heart, turns into a steady flow in capillaries in the periphery. So what's the name of this?
I wasn't listening, sorry.
Oh, you weren't listening. I thought you were referring to this. Anyway, so this whole thing, blood flow, can be studied quantitatively, and there's this Hagen-Poiseuille equation that kind of is the physiological equation. To express all of this quantitatively, I decided, when I designed this course some years ago, or it was kind of in the process of designing, not to go into this quantitative framework. Whatever one can, but we don't. So that was on blood flow. How can blood flow now be controlled in for the brain? And this is just essentially a repeat of what we already discussed. So there are two kinds of cerebral blood flow control. One is the central autoregulation control, which are the mechanisms of the autonomous nervous system that maintain a constant perfusion of the brain. So this is essentially how much blood goes to your head just by default, which is of course tightly regulated all the time. Sometimes when you have been laying down, relaxing for a time, and then stand up, you might, depending on your blood pressure, might realize that there's a little bit of, or there's too little blood going to your head for some time because you feel a little bit dizzy. So these kind of things, but then that's upregulated, and the proper pressure is applied, and then you're not dizzy anymore. That's autoregulation, but that's also, of course, cerebral blood flow control. What we are more interested in, of course, as we established in the very beginning, is functional hyperemia, which is this increase of blood flow in response to a local increase in neural activation. Yeah? So we are not that much interested in the central autoregulation of cerebral blood flow control, but this functional hyperemia level of control. This shows essentially something very similar to the Saracen and Roy example, but a little bit more in detail and a little bit more in terms of a functional perspective. So again, this is refers to a sciatic nerve stimulation experiment, this time in the rat. So this black line shows when the sciatic nerve was stimulated. Don't know whether I mentioned it previously, but the sciatic nerve is this big nerve that goes to your legs from your spinal cord and also in the red. So it's this big nerve that goes along your back, essentially, then to your into your legs, and this is being stimulated. And what they observe for a certain region of the brain, of course, is that the arterial diameter increase, which also then led to an increase in blood flow, while the blood pressure that was measured was actually the same throughout the experiment. So what this shows in terms of experimental evidence is that neural activity in the brain that is induced by the peripheral stimulation of a nerve changes the diameter of vessels in the brain, and this changing of vessel diameter in the brain then leads to an increase in blood flow. Yeah, it's something one can suspect, but this is then experimental evidence that this can actually be observed. Doesn't mean that it's the only process that can ever happen or something, but it definitely shows some experimental evidence that blood vessels change their diameter, and in this way the blood flow can be increased. So how can this now happen, and so how can, when there's this, if there's this covariation between blood vessel constriction or relaxation and neural activity, how is this now done? So how can this neural and vascular things interact? And there the Hüttel at least distinguishes, I think, 3 levels of blood flow control: extrinsic neurovascular control by central auditory regulation, and the neurons of the autonomous nervous system that modulate the vascular tone. Again, this is something that we've seen. And what is called intrinsic neurovascular control, inducing functional hyperemia, and being due to cortical neurons and astrocytes modulating the local vascular tone, and metabolic control, which is also related to functional hyperemia, and is not done by means of cells that are active, but by the byproducts of neural activation that modulate vascular tone. Yeah, so so these are now mechanisms of blood flow control that can either induce global changes or local changes, and the local changes can either be done by means of cells or by means of metabolic substances.
What exactly is this vascular tone?
So that means just the vascular tone is, how can I describe it? That's kind of the constriction level of vessels. So the vessels, the arteries, and then also the arterioles, so everything in the artery system is, there are muscle cells in the vessel walls, as some slides ago that were shown. These are smooth muscle cells, so they can constrict, and by constriction increase the tonus, so the Anspannung or whatever, so the tone,
Tension.
Yeah, the tension, exactly, in a vessel, and this can be modulated because these are smooth muscle cells which can constrict and not constrict. Yeah. So tone is, John's tone is here. It's basically the tension. Pressure. It's a tone. Yeah.
The English word is a little bit unlucky, I think. In German, it's tonus, doesn't really confuse with other words, but tone.
one says about persons, one says he or she is quite toned. One says things like that. It means something. But it's a little hard to explain what it actually means.
I would also struggle.
This shows just a picture of the things on the previous slide, and a little bit kind of the histology or microanatomy of this whole thing. So the idea is that there are these pile arteries which are primarily affected by extrinsic neurons, and there's some regulation of the tension of the tone of these vessels at this level of regulation. If you go for these intracortical arteries that we've seen before, as they go into the calcarine sulcus, or as they not go into the calcarine sulcus but are there at the calcarine sulcus and go into the cortex, they are kind of supplied by these astrocyte feet. And the astrocytes, again, being located also at the location of synapses. So you can see that here, for example, and synapses, of course, coming from neurons. So the idea is then that in the cortex, astrocytes play an important role. They sit at synapses, and they sit. They have their feet at these intracortical arteries and can contribute to the local blood flow control. And this then basically shows that in even more detail, so it zooms in more. So at the more capillary, is that correct? Yeah, at the more capillary level, you have things that you can call neurovascular units, being composed of a neuron, astrocyte, and pericytes. Pericytes are just these smooth muscle cells in small arteries / capillaries that have a different name. So they are pericytes. They can constrict. And this is what is shown here. So you have the capillary, you have the endothelial cell, so one cell that makes up the wall of this vessel. Around it you have the pericyte, which is essentially one layer of smooth muscle cell which can do some constriction. And then you have the astrocyte here, so the astrocyte end foot. And in the vicinity you also have kind of neurons and their synapses, yeah? So that you have this unit of things that are actually nervous tissue and relate to the brain, and histological entities, astrocytes, pericytes, and endothelial cells that make up the vascular system, yeah? And this shows the anatomy at the microscopic level, but also suggests that you can have quite close interaction there. For example, by neural activation, inducing astrocyte calcium influx, and then the release of vasoactive substances that lead to a constriction. Yeah? So the idea is that this is that it's not like so maybe to emphasize. For me, it's like that's how it is. It's interesting. And it's not somehow surprising, but it could be different. So I think this is the point here. That it could be different, that, for example, you have just the artery, and this can constrict or not constrict, and then there is no regulation possible anymore inside the cortex. Yeah, so that regulation just cannot happen there because these constricting cell types are not there, and everything that is can happen in terms of the spatial regulation must happen at a larger level of arteries, meaning that the spatial specificity cannot be that strong. Yeah. So this is how I think one can see these things and appreciate them. Otherwise, it's a little bit like, oh, they are cells. Good. So I think one has to think about the alternative. Then it's a little bit more surprising. Good. So this was now a lot on kind of basic energy consumption, cerebral vascular system, and cerebral blood flow control. Now getting a little bit more closer to what we are more interested in, actually, namely the Bolt signal, the hemodynamic response, and how this is related. But these things that we established until now will come up again. So this is why we discussed them. So Bolt signal. Again, Bolt signal, as you already know, has to do somehow something with oxygenated and deoxygenated blood. We briefly mentioned that. And it has also something to do with an increase of blood flow locally. This is what this whole story until now was about. Now we're trying to put these two things together. What this graph shows is kind of also the history of the Bolt signal, if you want, or the history of the idea that somehow oxygenated and deoxygenated blood have different magnetic properties. So this shows very early on that Ravi started kind of the nuclear magnetic moment measurements that we discussed and will discuss again. But at the same time, or briefly afterwards, Pauling, so Linus Pauling, the guy with vitamin C and cold, which is not true, but it's still Linus Pauling. He also did a lot of other things in terms of the DNA, RNA business. They studied the magnetic properties of blood, so that deoxygenated and oxygenated blood have different magnetic properties. That was also kind of seen in parallel with the development of magnetic resonance as a thing, of course, a long way before it was used for imaging. So then more nuclear magnetic resonance before the war. Then some development going on in nuclear magnetic imaging. So some things that we talk about, that we have talked about, for example, relaxation times, but also using MR, using gradients to make images, echoplanar imaging. And then kind of in the early 80s, the effects of blood oxygenation on T2 star contrast. So these time constant of the transversal relaxation. So kind of a link between blood oxygenation and measurements that you can get from MR. And basically this being supplemented throughout the 80s. And then the kind of crucial breakthrough or whatever in 1990 by Ogawa to use the Bolt contrast at basis 5 MI. That's kind of the history, but again, the history should, or what I'm trying to emphasize, not the Hüttel of course emphasizes, is that it's not that everything was discovered in 1990, that suddenly it was known that blood and oxygenated, deoxygenated blood changes magnetic signals. That was known for a long time. It was just more doable, and it came together then in 1990 on the shoulders of giants. Now that we talk about Bolt signal, we need to talk a little bit more about blood and how blood works and how hemoglobin works. This just shows one thing. So we are only actually interested in erythrocytes. But I worked with a computer scientist early on in my PhD, and coming from medicine, I always had these pictures of erythrocytes in my head and how they, just from cartoons from on the body and whatever, how this happens, and I figured that the computer scientist actually wasn't aware of these. Of course, I wasn't also aware of a lot of things in computer science, clearly, but then it felt somehow strange not to know about erythrocytes, but analyzing fMRI data. On the other hand, it's also very strange to analyze fMRI data and not knowing anything about the Fourier transform. So it goes both ways. And this is why I included this graph, so that everyone knows about erythrocytes. They start as cells that actually have a cell body, a cell nucleus, so a cell kern nucleus, but they eject their nucleus at one point. So 30 hours into their life, they decrease their RNA quite a lot, and basically just consists of a lot of hemoglobin, which is a protein, and then are released into the bloodstream and live for how many days? I think 120 days. That's somehow what I remember, on average. So not forever, so that means that your blood on average is exchanged every 4 months, every 4 months. I think that's something I remember from medicine. So it's constantly being replaced. Now zooming in even further, these are erythrocytes. Within the erythrocytes you have hemoglobin, and hemoglobin is a molecule which consists of 4 chains, and change meaning change of amino acids that make up proteins. So there are these 2 alpha change, beta change, not really important. What they also comprise are, I think, 4 heme units, which are these kind of molecules which contain iron. So this is why you need iron, and which can bind the oxygen. So this shows why you need iron if you have problems with your blood, and it shows that hemoglobin is a molecule that consists of a protein and these heme units. Now I think what is actually more important is this cartoon here from Wikipedia, which shows that the oxy and the deoxy hemoglobin have different spatial configurations. What you see here are these alpha and beta chains, and you see these heme units. There are these hemes. What you don't see is how they bind the oxygen molecule. So the oxygen molecule is bound here, but it's not shown. What is, however, shown is that if it's in the oxy or the deoxy state, the configuration of the heme units with respect to each other changes. And the idea is that this changes the magnetic properties of the hemoglobin. So the deoxyhemoglobin, so if the spatial configuration of the heme units is a certain way, then the thing is paramagnetic, and it affects the magnetic field and induces signal loss. If it's in the oxyhemoglobin configuration, then it's diamagnetic and doesn't affect the MR signal, so it doesn't have an effect on the local magnetic field. Yeah, so that's kind of the microanatomy story of why oxy and deoxygenated hemoglobin affect the magnetic field. Of course, you could now start asking, so why does a certain spatial configuration of heme units affect a magnetic field and another doesn't? And that's then where you hit the bottom of this course. That's the level to which we go. We don't go any deeper. But it's an interesting question. Now, what this shows are the early results by Thalborn that now relate blood oxygenation to relaxation time in MR. So let's see that I get this right. So this shows measurements of T2 time constants, and this shows measurements of T1 time constants, and this shows on the x-axis an increase in blood oxygenation. The T2 time constant is lower for higher levels of blood oxygenation. And if you transfer this into an image, you have a signal dropout for high oxygenation. This we will see in a second. The important thing on this, because it can be that it's this way around or this way around, the important thing is that the T2 time constant is clearly modulated by blood oxygen. So you see that for a certain level of blood oxygen, it's different than for another level of blood oxygen, while the T1 time constant is not modulated by that. Yeah. So the point of this slide is that the T2 time constant, T2 star time constant that we discussed previously is affected by blood oxygenation. T1 time constant is not. So we're interested in T2. This is easier in terms of remembering which way it goes. So this is from the original Ogawa experiments from 1990. This used a T2 sensitive imaging procedure for rats, and there was animal MRI. There will be a talk on animal MRI next week in the CCMB seminar. This shows the brain of the rat imaged by MRI when it's breathing 100% oxygen, so it has full saturation of its blood. And here you see if it's breathing normal air. And what you should see here is that there are more of these black things. So here you have a little bit black stuff, but here you have definitely more black stuff. So you have these black intracortical things. They are all black. And black meaning signal dropout. And so the idea is that the signal dropout somehow is related to the oxygenation level of the blood of the rat. Because here it's only getting oxygen, here it's getting normal air. Now, to also verify this not only in vivo, this association, but also in vitro again, what they also looked at is oxygenated hemoglobin in a test tube under two imaging procedures with a spin echo and gradient echo, and deoxygenated hemoglobin, where you definitely in the gradient echo sequence see more kind of a irregularity, let's say, and also with the spin echo you see a little bit irregularity. And this was essentially their confirmation that these changes in the images that you see here and here have to do really with the oxygenation level of the hemoglobin, which here of course is only indirectly assessed because it could be something else that is induced by the breathing of oxygen versus normal air. Yeah? So the bottom line again is if you have deoxygenated hemoglobin somewhere, there's signal dropout. That's the kind of essential message that deoxygenated hemoglobin co-varies with signal dropout. And we've seen that in a different format or in different contexts already. So based on this Ogawa experiment, Ogawa himself and Peter Banditini, I think we will see some very recent work of Peter Banditini at a later point in this course, because I want to include something on layer-specific imaging, and they just have a new preprint on layer specificity. So he's been around for a long time. Well, he's actually quite toned. I just saw him at the CCN conference, and he was running on the treadmill while I was doing something else. Anyway, so that's Peter Banditini. So they basically following up on the Ogawa lead, where basically this was done due to breathing oxygen. They then, of course, made the connection, and this was also discussed in the Ogawa paper, that if neural activity somehow changes in blood flow, this must somehow also change the oxygenation level. And this can be induced by neural activity. So these are these early experiments on fMRI that show that if you stimulate a participant using light, that then the signal, the MR signal in the visual cortex goes up. Yeah, and this really works. This has been replicated many times. I have replicated it myself. Everyone has done motor response as well. So we've seen that before, and now a little bit more in detail. One thing to mention here is that these were what we would now call block designs. So they put on the visual stimulus for a long time, almost a minute, and then they had it off for a minute. What was also shown, and that's nice that it's in the middle, but which was often forgotten, is that also very brief stimuli can increase the bolt signal, and this is much more of what we are doing right now with fMRI, because we have participants do tasks where the typical stimulus and change of tasks and so on is more on a multiple seconds level and not on multiple minutes level, which was due to the earlier work on PET that they didn't this long. And this was also shown already in the early 90s, sometimes forgotten. But coming back to now the hemodynamic response and this idea to relate now everything together. So what this story was about the bolt signal was more that it's on its own is an interesting effect that the blood oxygenation level affects MR signals, and that has been discovered and demonstrated. But again, this is, if you want kind of the psychology language there, this is more like correlation, and the question is, why does that happen? So why is there this link? And this is now finally where we're getting to. Is there a question?
Yeah, I'm sorry to one of the slides before. I didn't get this change from oxyhemoglobin to deoxyhemoglobin. This one. I didn't get it at all.
This just so the point here is that there are these heme group which have iron molecules in them, so they have some electrical magnetic properties because they have these ions in them. Yeah, and then the fundamental idea is that the configuration of this electromagnetic entities is changed when the when the hemoglobin binds oxygen versus it doesn't. So what you need to look at is here at the right side. It says oxy-deoxy. Maybe it goes a little bit unnoticed. And the other thing that you of course see quite a lot is that the spatial configuration changes, including the relative position of this heme group. And yeah, so the point is just that these different magnetic properties of the hemoglobin molecule are due to a change in the spatial configuration of the whole molecule, including the heme groups, under oxygen binding versus not oxygen binding. And so the idea is simply, so you can basically say, so let's say you can also, as an analogy, you can have, okay, you have two magnets, so two magnetic active things, and if you put them into a certain spatial configuration, they cancel each other out in terms of their external effects. Yeah, so think that somehow you set up two magnets in a way that outside of these two magnets you don't measure any magnetic activity or magnetic field. Now you change the spatial configuration of these two magnets by, I don't know, twisting them, and then you measure a magnetic field outside of this configuration. So that's kind of the story. Yeah, of course the story is only works to a certain level of depth, because then you go into the physics and one would have to discuss, so what are exactly the magnetic properties of these heme parts of the hemoglobin molecule, and how does the configuration change that it's now really trivial to see that this change in configuration affects the external magnetic field, of course, and this is not discussed, but maybe that's what you're after. So this would be then kind of a electromagnetics in 3D physics exercise. Yeah? Other questions? No. Good. So now I'm trying to tie it all together in terms of the hemodynamic response. So first, in terms of the bold idea, so the bold idea is that in the resting state, the level of oxygenated and deoxygenated hemoglobin is maybe roughly similar, which induces signal loss. Now, when the brain gets activated, so when neural activity happens locally, then locally there's an increase of oxygenated over deoxygenated hemoglobin, which leads to less MR signal loss and hence an increase in the MR signal. And this is kind of the most complicated thing of this whole thing, although it's not really that complicated. It's just the other way around as one would expect. So what has been observed always is that neural activity, but also oxygenated hemoglobin makes the MR signal, the T2 weighted MR signal go up. And now what this shows is essentially a little bit more in detail why this is happening. So the active state, in the active state, there is more oxygenated hemoglobin than deoxygenated hemoglobin, hence less MR signal loss, hence a higher MR signal. Image gets brighter, essentially. And the surprising thing from the whole theory even before is that now there's neural activity, suddenly there's more oxygenated hemoglobin, because one would, if one had the chance to think about it, one would have thought, okay, active state, neural energy is required, now the oxygen is eaten, so that there's less oxygenated hemoglobin. However, the opposite is the case, there is more oxygenated hemoglobin. And the basic explanation for the whole thing is that it's kind of an overregulation. So yes, there is energy demand, and yes, oxygen gets eaten, but it's made sure that enough blood is there, so nothing bad happens. So there's an oversupply of oxygenated hemoglobin. And then the question is, in terms of this metabolic hypothesis of functional hyperemia, how does that happen? And this is kind of then zooming in on the details of why that happens. So this is this important uncoupling of cerebral blood flow, cerebral metabolic rate of glucose, and cerebral metabolic rate of oxygen. Yeah, I think this is quite important. So please learn this for your exam. I had a lot of people last year that they could talk about a lot of things, but somehow this got lost. But maybe because the slide is not that fancy or something, or there are these weird abbreviations. But this is actually a little bit at the heart of the whole thing. So the question is now, okay, bolt signal goes up when there is energy demand, when there is neural activity. But why is this the case? And the answer is that there is an uncoupling of cerebral blood flow and the cerebral metabolic rate of glucose and oxygen. So what are these things? So cerebral blood flow is just how much blood flows through a certain area of the brain. And during activation, the cerebral blood flow goes up. This is what we would expect from everything that we discussed previously. So more blood rushes into something. What is also the case is that there's a higher rate of glucose metabolism. So glucose is eaten up. So 51% increase in the eating or the processing of glucose. That also makes sense. Activation needs energy, so you eat more glucose. What doesn't hold up with this is how much oxygen is actually extracted. So the oxygen, or how much is metabolized. So the oxygen metabolism only goes up by 5%, which is not much. The other ones double. They increase by half, so they get to 50% more, and the rate of oxygen metabolism only goes up 5%, and this is this uncoupling. And of course, this immediately, if you think blood flow goes up and glucose goes up, but oxygen does not go up, oxygen extraction or oxygen metabolism does not go up, means that there is more oxygenated blood. But the question is still, why is this uncoupling happening? But that this uncoupling is happening, this is quite important. And this is also, the reference of this paper is also in the Hütte. Now, there are many theories of why this is happening. And one theory that is, in my opinion, not really understandable, formulated in the Hütte, So I try to kind of come up with my own theory of what this is supposed to mean. Is this astrocyte neuron lactate shuttle theory, which is a theory in itself. I know that you're reading a lot of summary papers in the other courses, so some reviews. You can also find these reviews on the astrocyte neuron lactate shuttle, and these reviews are written like the reviews that you are now reading, right? So there's a little bit of thing that makes sense, and then there's there are many paragraphs where they discuss these people did this and this and found this and this, and once these paragraphs start, you get a little bit lost, and it's a little bit annoying to read, but then in the end it comes together, and yes, and this is now the general theory of anhedonia and I don't know, amygdala, whatever. And at this level of theory, if you know what I mean, this astrocyte neuron lactate shuttle theory exists in the literature. Yeah? So it's not something that is really that clear cut. But the idea here is that neural activity happens. And of course, we have seen that neurons and astrocytes are quite closely together. Astrocytes have to do something when neural activity happens. So they are involved quite a lot in synaptic activity, as we've heard before. That means that they themselves also need energy. And now the point of this whole thing, although the name doesn't really give away the point is that the astrocytes don't have mitochondria. Hence, these astrocytes, if they become active locally, they rely on anaerobic metabolism. So they actually need glucose, but they don't need oxygen. They become active, and this is one problem, hypothesis about why this uncoupling takes place. So the idea is it's the astrocytes that need glucose, and they need hence they need more blood to a certain area, but they only need the glucose, they don't need the oxygen, and this is why you have this oversupply of oxygenated hemoglobin, which induces this increase in the MR signal. It's one theory. There are and this is the only one that I go a little bit into detail. There are other explanations why this uncoupling happens and why oxygenated hemoglobin is more prevalent than deoxygenated hemoglobin under activations. And these other theories are the following. Maybe it's not only the astrocytes, but it's in general an increase in the anaerobic glycolysis for quick ATP production, so that all the cells in a certain area, including the neurons, they feel like, wow, we are active, we need quick ATP, let's do anaerobic glycolysis, let's eat up glucose, but let's not bother with the mitochondrial electron chain so that we quickly get some ATP. That's one, and of course these theories they also will be backed up by experimental evidence. Yeah? So I'm discussing them here as theories, but of course there are papers that show that something like this is happening. Maybe it's simpler. It doesn't really go via metabolism in this case, but it's due to the fact that if the blood flow increases, then the extraction of oxygen is worse, essentially because the blood rushes by faster, and these diffusion processes, they don't work that well anymore. So this is why the oxygenated hemoglobin is increasing under activation. Experimental evidence for this exists, but there also exists experimental evidence for other things. Maybe it's it's actually what is described here. It's a little more intricate in the sense that where this increase in oxygenated hemoglobin is seen, this is actually not the metabolically active region, but the metabolically active region is further downstream, and this perfusion is it basically just goes through there. So it's actually the metabolic active region is a little bit downstream. So this, I guess somebody has done in an experiment where they saw, oh, this is more metabolically active, and here we see an increase in oxygenated. So this is could be the case. Maybe it's actually also a safety mechanism. So if you think there's activity and you go further downstream in the capillary arteries, vascular system, arteries, capillaries, the aim is to make sure that while there is an overabundance of oxygenated hemoglobin in an active area, that this makes sure that the distal tissue, which is downstream, still gets enough oxygen under these increased metabolic circumstances. Yeah, and overshoot only for tissue in supplying arterial vicinity. I think that was last year the same thing, that I didn't really know what this last part is supposed to mean. Overshoot only for tissue in supplying arterial vicinity. Well, yeah. Maybe let's keep it at this. So these are now, because these are already 4 ideas of why this is happening, and then the 5th was on the slide before, that's mainly the astrocytes. And the thing is then that kind of the verdict is still out there in a way, at least from the Hüttel book. Like it always is, I would assume that all of these things contribute, and some of them are contributing more and others contribute maybe a little less. But yeah, it's not only one simple mechanism, like often in biology, from my perspective.
I didn't quite understand the difference between the last 2.
Yeah, so this would be that would be basically so this would say so the question is, where is the metabolically active region? So in this under this theory, the metabolic active region is downstream, and what one sees in the region where one sees this increase in oxygenated hemoglobin is superfluous. This goes different here. The metabolic active region is the region that one sees this increase in oxygenated hemoglobin, and although it's too high there, it's done because actually there's oxygen being metabolized in this active region, and this over-perfusion is just to keep distal tissue, which is not metabolically active, safe in terms of that it gets its usual oxygen. That's kind of the difference. Yeah. So I think I don't know how many pages in the Hüttel book that I condensed onto this, but it's we have to stop. We actually have to stop because I have to also go somewhere. It's different theories why you have this uncoupling. Yeah, so it's important that you keep in mind that there's this uncoupling, and that there are different theories of why this uncoupling happens. And I would then suggest that we stop right now and discuss the remaining stuff next time, because I have to some at 12. Good, then let's just stop, and if there are questions, keep them for next time, then we can discuss them.